we're going to continue now, and our next speaker is Emanuel Petricoin, but actually he goes by the name of Chip. And if you hear his talk, you will realize that that's actually a very apt name for him, because he has been involved in chips for proteins, chips for RNA, for a lot of his working life. Started out working on Neisseria, and has gone on from there. And actually, with Lance Liotta, was responsible, I think I'm right in saying this, before I joined SAIC Frederick, there was a bit of a furore at the NIH, if you remember, because they were banned from getting involved in any commercial activities at all. Even being bought coffee by a company was banned, still is, as far as I know. And it was thanks to Chip and Lance that actually happened. And it's one of the most short-sighted things that has ever been done, in my opinion, because it's stifled innovation in the national health here for a long time. But no more ado, please come and tell us a little bit about what you've been doing.
Thank you. So thanks a lot. It's great to be here. Thank you very much to Larry and the organizers for inviting me. Larry and I have had a great kind of orbiting friendship over the years and have really had some interesting times where we've been able to speak together at really fascinating meetings. We were at Princess Takamatsu cancer meeting a while ago, and that was fascinating, and then had the surreal meeting with Bill Gates a couple years ago, and when Larry and myself and I think Rudy Aebersold went up, and that was a lot of fun. So every time that I see Larry, it's always at these very interesting meetings. And one of the things I always felt with Larry was I've never seen someone having so much fun doing so much important work. So I really look up to you for that. So I wanted to talk about, there's so much interest, right, in genomics and precision medicine and the Cancer Moonshot Project. And I don't know about you, but in DC, every minute or so, it seems like a commercial on TV about precision medicine and Cancer Treatment Centers of America commercials, all sorts of commercials. But the field of proteomics is kind of dragging behind genomics right now as far as the federal funding. But there's groups, SomaLogic being one of them, that have been developing tools to look at proteins for a while because of their relative importance in biology, is sometimes overlooked, interestingly enough. And I wanted to cast light on a couple different case studies that I thought may help to kind of put this into a proper light. So, somewhat self-evident thing to say, but let's start out. DNA, the blueprints of the cell, but it's really the proteins that do the work. If you want to think about it more conceptually from a computer standpoint, it's really the DNA is the hardware of the cell, but it's the proteins are the software of the cell. And actually, interestingly enough, as I spent about 15 years at the FDA, I realized that most of the biomarkers that are FDA cleared, that perhaps we have measured in our own physicals, checkups, things like ALT and SGPT and CRP and PSA, those are all proteins. And I'll talk about this a little bit. If you look on section 12 of the package insert of your prescription medicines, it tells you about how the drugs work. Most of the time, the mechanism of action of the drugs is actually against a protein. And many times, the therapy itself is a protein. Things like interferons and interleukins, right, TNF. So sometimes the therapy is a protein. Many times, the mechanism of action of the drugs are against proteins. And many, many times, the biomarkers we're using to select patients or to monitor for disease are proteins. And this is being revealed really in the field of cancer in the past couple years with the elucidation of many of the cancer genomes through the Cancer Genome Atlas Project, the TCGA initiative, where they're cataloging and describing the entire genomic content of cancers. One of the biggest aha moments that's come out of that foray is the heterogeneity of cancer at the individual level. Remarkably, everyone's cancer is unique. And it's filled with a hodgepodge of mutations and alterations, only a handful of which are really driving the cancer. So one of the problems with cancer, right, is the underpinning DNA instability and the DNA mismatch repair systems that have gone awry, and they build up mutations, some of which become causally important to tumor genesis and pathogenesis and metastatic progression, and a number of them are of no consequence and are happening because of the underpinning derangements. So you get this Tower of Babel information, right, when you sequence a person's genome from, say, a colorectal cancer tumor specimen and another patient's colorectal cancer tumor specimen. It looks entirely different. And this probably explains why most chemotherapeutics don't work and why handfuls of patients have to be selected to identify a specific driving event for a specific drug. And one of the things that occurred when they were doing the bioinformatic analysis of this kind of Tower of Babel was that if you started to understand the genomic mishaps in the context of the proteome, then it started to make more sense. So one tumor may have a mutation in the gene PIK3CA, and another tumor may have a mutation in PTEN, So the names of these were ones where you could start to understand that biochemically at the enzyme level, they form pathways. And when you hear about pathways in cancer, don't think genes. Genes don't form pathways. Proteins form pathways. Proteins are the effectors. They're the enzymes of the cell. Many times in cancer we have, and most of the no-targeted therapies, are enzyme modulating inhibitors, kinase inhibitors, for example. Because these pathways that most patients share are really at the protein level. Now, of course, a lot of molecular biologists have these kinds of posters up all over their labs. There's ones like from cell signaling, they're kind of infamous, right? That are just kind of hanging around every single quarter. There's probably 30,000 of them in Building 10 at the NIH, right? They're just all over the place. And they're cartoon representations of kind of the knowledge that we're understanding at these pathway levels. And, these are not two-dimensional maps. This is a hairball, and it's a fluctuating information archive. Which is why we're not likely going to have some Science and Nature series that talks about the elucidation of the human proteome, because the human proteome is really a fluctuating information archive. And it's even cell type specific. And perhaps even in the context of that human being, what that human being's not only genetic background is, but what they ate that day, and the whole constellation of activities that make us human beings actually is reflected in our proteome. Now, what's interesting to us is that these pathways, a number of them are extremely important in cancer. And you talk to someone like Rob Weinberg, he said there's only so many ways that you can get cancer at the molecular level. There's not an infinite number of possibilities. Cancer is a proteomic disease at the functional level because it's the proteome as a software of the cell. The genomic mutations that you get are selected for by whether or not the proteins that are encoded by them make a difference to the cell's ability to survive, adapt, move, and intravasate and extravasate in and out of the bloodstream and lymphatics. So we want to be able to understand these pathways better. And proteomics is perfect for this because you can provide a direct measurement of these pathways. Now, the majority of these pathways are regulated by a series of modifications that themselves are regulated by proteins. One of the most important protein modifications is phosphorylation, which are put onto proteins by proteins, kinases, and are taken off of proteins by proteins, phosphatases. And it's this yin-yang of phosphorylation, dephosphorylation, which sets the threshold of whether or not a protein is activated or not, because these phosphate groups allow proteins to interact, and by interacting, allows them to, with their dance partner perhaps, modify their dance partner, and that dance partner goes on to modify a different dance partner. And this is how cells communicate, inside and outside. And in cancer, this process is hijacked many times. So 2 tumor specimens under the microscope would look identical. And those 2 patients do extremely differently in a clinical trial given the same drug. So histologically, the way that we pathologically describe cancer is inadequate to explain what we see when we give a patient to therapy very often. And so conceptually, if we could find ways of measuring these pathways ahead of time that are activated, it could provide new information beyond what the genome itself describes for those patients. And this is kind of the era of multi-omic analysis, where you are profiling these patients at the genomic level, you're profiling them perhaps at the phosphoprotein level and protein level, and the aggregate of that information, like the blind man and the elephant analogy, you start to look at, hey, here's the trunk, here's the leg, I'm dealing with an elephant, and I'm describing cancer better now than I have before. And this is the hope. And it's, as I said, it's important to recognize that proteins are already important, guys. They're already important because we have drugs, as I said, that work against the protein. And I encourage you to look at your package inserts every once in a while. A lot of people just throw those away. When I was at the FDA, we spent a lot of time on the wording in those package inserts to make sure that the drug companies were describing adequately what they found in their clinical trial and what they know about their drug. So this is a drug called Afinitor. I skipped over. So this was approved for breast cancer a little while ago for ER-positive tumors. And this drug, if you look on the section 12, clinical pharmacology, it says what the drug does. Is an inhibitor of mTOR protein, a serine threonine kinase enzyme downstream of this pathway. And it says basically this drug binds to this protein, resulting in, you can read it, but the point is I don't really see a lot about genomics here. I see a lot about what the protein target is and what the drug does to this protein and how it modifies it and it influences enzymatic activity. Now I could throw up example after example of this kind of result. And so philosophically, it's important to study the proteome of cells. It is a direct readout many times of the mechanism of action of these drugs. However, there's a lot of technological barriers for proteomics, huge number. One of which, especially in the context of looking at the signaling circuitry, is, well, the tissue input to most molecular profiling is very small. 80% now of the trials that are biomarker driven in oncology, the input specimen is an 18-gauge core needle biopsy worth of material, and that's it. And the IRBs don't allow you to come up with a technology to fit, or they're not going to allow you to take hundreds of punch biopsies of a patient's breast cancer because that's what your technology needs. You have to find a technology to fit the very small amount of material that's coming out of these clinical trials. There's also a great deal of anxiety about the pre-analytical variables of tissue fixation and time to preservation. The proteome is a fluctuating information archive. It's not set in stone like the genome, and so we have to be careful about how long the tissue sits out on the bench, how long the blood sits out on the bench, how long it was freeze-thawed. All these things impact the proteome. And of course, there's a tissue heterogeneity issue. And the end user, oncologists, want to go beyond whether or not a tumor is HER2 positive or negative or ER positive or ER negative. They really want a multiplexed panel of information about the cancer because they want to figure out which drugs to give. And there's a constellation now of drugs that could be considered. Even more pressing is the fact that, as much as I'm a proteomophile, there's a lot of people that want access to the same piece of tissue. In these clinical trials, you have a whole series of technologies that are sitting there like little birds in the nest, waiting to be fed the tissue coming back from the trial. So we're competing for tissue. And as I say, tissue is the issue. In these trials, you have to be very cognizant of the fact that you're just one of many mouths to feed. So in the face of that, and there is no PCR-like technology for proteomics yet, and so there's no way to amplify endogenously protein copy numbers so that then you can just see these low abundance proteins with existing technologies. So you have to kind of build almost like a Rube Goldberg mousetrap kind of technological daisy-chained workflow to be able to get at some of the low abundance proteins that are sometimes driving cancer. So about 15 years ago, kind of faced with the small amounts of material that we were having to work with and really wanted to explore the signaling circuitry of the cell, we invented a technology called the reverse phase protein array, which is now somewhat widely used, but it's really kind of a nano dot blot technique that allows us to take very small amounts of material and multiplex the analysis of many specific phosphorylation signaling events so that we can kind of measure each of those hubs in that circuit map that I showed you. So what we can ostensibly do is from a tiny little core needle biopsy, we can measure quantitatively hundreds of phosphorylation events simultaneously. Now this is a profiling-based technique because we're kind of looking at 300 lampposts, right? We're only able to look at what we postulate is important to look at. And each of these requires a specific antibody. And when Larry and SomaLogic start getting aptamers to be able to find these types of modifications, we can use these technologies as well. So we're looking at whether or not these proteins are activated in very small amounts of material with this technology, and we have graduated this over the past decade or so from an idea to now CAP-CLIA accredited workflows and a commercialized test that our physicians can send the tumor in and get in 10 days a snapshot of the signaling circuitry in that patient's tumor. Now I want to showcase one specific event that occurred in the last year that I think really amplifies on the need to do this. There's nothing like showing you an example where if you didn't do this, you would have missed out, especially in the clinic. And what I'm going to show you is the end result of a journey that we now think will result in half of the women with triple-negative breast cancer being effectively treated with a drug that wasn't designed for them. So one trial that I'm participating in is the I-SPY2 trial. It's a pretty well-known adaptive design trial for women with breast cancer. And in this trial, which is one of the world's first adaptive design trials, which I'll show you about, drugs are being cycled in and competed against each other in what's called the neoadjuvant setting. The neoadjuvant setting is a time period between diagnosis and surgery. And what they found, what the FDA found in these trials with the pharmaceutical companies is, in fact, in this neoadjuvant setting, this time period between diagnosis and surgery, that instead of taking a woman to surgery right away, you can give them drugs that will shrink the tumor prior to surgery. Now, initially, the thought was, well, this would be maybe breast-sparing surgery, and the surgical time will be reduced. The amount of tissue taken out will be reduced, and so you will end up having better cosmetic results. And that's the case. But what really threw it over was the fact that when they followed these women over time, their overall survival was benefiting by shrinking the tumors in this setting. And this is why the FDA has really allowed somewhat of the paradoxical aspect of being diagnosed with cancer and not immediately doing something, but saying, okay, we're going to give you drugs, shrink it, go to surgery, smaller tumors, better outcome. Well, now in this setting, we can use this as a sandbox, a molecular sandbox. And it provides proteomic scientists and genomic scientists an opportunity to get longitudinal samples from a woman before treatment and during the treatment schema. And this is what is done in this trial. And in fact, it's much more complicated than that in this trial because you have, a variety of agents that can be competed against each other, and they actually look at MRI images, high-definition MRI images during the course of the trial, and they use the reduction in the kinetics, the actual differential equation of volume over time, to decide the enrollment criteria for the next round of therapy. Basically, what I'm saying is it's a voted weighted average of which drugs are given to which woman. If a drug is doing well, more women get the drug. If it's not doing well, less women get the drug. And they can actually kick to the curb drugs that are showing futility and graduate drugs that are working well. And that basically is done during the course of this therapy. In this trial, we give drugs to a whole cadre of the whole spectrum of the molecular architecture of tumors as they come in. HER2 positive, HER2 negative, ER positive, ER negative. And this will become important because, to cut to the chase, what I'm going to show you is triple negative breast cancers. As I said, there's a specific subset we think we can help because of this trial results. And I'm going to get to that. So here's a snapshot of some of the drugs that are in the trial. This is actually a little bit old. I was working on trying to get a newer slide because we're up to 17 drugs now that are in this trial. All different types of pharma companies' drugs are in this trial. I'm going to concentrate on one of the drugs, neratinib, which is made by Puma Pharmaceuticals. And it has as its mechanism of action a kinase inhibitor against HER2 and EGFR, two important proteins that drive a lot of cancers. And these two proteins are found on the surface of breast cancer cells in a select subset of women. These specific drug targets on the surface are the HER family, very important family, that bind a variety of ligands that bind to these under normal circumstances and transmit a signal. And these are all happening downstream. And here are the two proteins phosphorylated, and when they're phosphorylated, they come together. So neratinib blocks EGFR and HER2 kinase activity. So we speculated that women who will respond to neratinib will have higher levels of these two proteins on their tumors. Basically, our postulate was, if the drug's going to work, it's going to work against women who actually are using the proteins and are activated. If the women do not have these proteins and are not activated, the drug won't work. So that was our hypothesis. And when we looked at the data, we actually found that we were right. So that we presented this work at ASCO a couple years ago. So we look at who responded, who achieved pathologic response. When I say pathologic response, I mean the tumor was gone at the time of surgery versus patients who did not respond at all. And we looked and we said, okay, these little Ys and Ts, these are the phosphorylation sites that we're monitoring. So we're asking not only is the protein there, but is it activated? And in fact, patients who responded to the drug had higher levels of the two proteins at different sites, EGFR and HER2. Hypothesis was correct, and Puma was very excited by this because you ostensibly have found a companion diagnostic marker for their drug. And that was important because their drug graduated in a subset of women who are HER2 positive and HR negative. They had 55% of the women that were given the drug responded. And 32% of the women that were given a control placebo, or in this instance, a predicate chemotherapy, responded. And these are the statistical output from the trial. Now, when we looked at the biomarker positive, these phosphorylated EGFR HER2 patients, we actually got an even better result. So it was 55% versus 32%, and in our selected population, 67% of the women that were phosphorylated positive received benefit versus 20% in the control group. But that's not the real story. This is in HER2-positive patients. So when we looked at the level of HER2 compared to what we were getting on the array, we saw something interesting. And in general, what we saw was we had, first of all, good concordance. This is the quantitative HER2 measurements we're seeing on these patients, and it kind of explodes here. The green triangles are patients who are HER2 positive by an FDA-approved means of measuring HER2. So basically, all the women that were HER2 positive, we're seeing high amounts of that. And all the patients that were HER2 negative were all low. And we had some disconcordances, and some of these were even at the differences between what's a test called FISH versus IHC, which there's some wobble there as well. But in general, with no false positives, we had about 93% concordance. Okay, so it says reverse phase array data that we're generating is accurate. We could probably go to the FDA and get a 510K with the reverse phase array compared to HER2. But who would want to do that because you already got a good test? However, we were able to check a box accuracy. Then we looked at a test method called FISH, which actually looks for how much the copy number of that HER2 gene there is in the genome. And in patients that are positive, a lot of times that's because of gene copy amplification. That's why they become positive and addicted to this pathway. So as the ratio of HER2 to a housekeeping protein goes up, you have two alleles, so anything above 2 means you're amplifying the copy number. So you see these patients in green that are HER2 amplified have greater than 2, and patients under 2 are all HER2 unamplified. And here's our reverse phase array quantitative. And in general, again, those patients that had high amounts of HER2 by FISH were high by the reverse phase array. And in general, we had about a 95% concordance. Now, the surprise result was so take a look at this keep this population in mind. These are patients that are low for HER2. Low by FISH, low by IHC, low by reverse phase array. They're low. These women will never, ever, under current standard of care, be given a HER2-directed therapy. All of these women will. It is a fork in the road. Get HER2 therapy, do not. So when we looked, now not at the HER2 levels by total protein, but by phosphorylation level, we found a subset of these patients popped up that were HER2 negative by everywhere, which means you want to measure them, but they were actually activated, paradoxical. Because supposedly they were low or near 0, but yet as much as they were low, what was there was as activated as the group of women that were all HER2 positive. In fact, these are statistically insignificantly different so again, none of these women get HER2 therapy. They're all missed. All these women do. So we confirmed this in a paper, and it led us down this road that I want to tell you about. So these are women that are phospho-HER2 positive, missed, HER2 negative. So he said, oh, this is interesting, because if you look at breast cancer therapy, breast cancer in general, these groups of women here, these women with triple negative disease that are HER2 negative, they're also ER negative, we do not know what to do with these women. We do not know what to do. In general, they have an increased germline BRCA mutation. In general, there may be some risk factors, higher increased incidence in African American women who do not breastfeed. They're sensitive initially to standard chemotherapy, but they end up basically progressing rapidly out of that and die quickly. And it's a very heterogeneous disease. And these Kaplan-Meier plots, which basically plot the overall survival, progression-free survival of these women over time, are pretty clear that women who are triple negative do not do as well. So there's an urgent need to find therapies to treat these women. Now, neratinib was actually given to triple negative breast cancer patients. And we said, okay, we just learned this lesson that we have this group of women who are HER2 negative but have phospho-HER2. So when we go back and we measure all these proteins in these tumor biopsy samples from iSpy, we're going to predict that phosphorylation of HER2 and EGFR predict response even in the triple negatives. So we unblinded the data, and guess what? that was exactly the case. The only two proteins that predicted response to neratinib in triple-negative breast cancers were whether or not the women had activated HER2 and EGFR. So much the case, and this is a two-way scatter plot, that's actually plotting phosphorylation of EGFR and phosphorylation of HER2. So the women that had both in green are the women who achieved pathologic response, and the women in red who did not. This is the treatment group and the control group. So all these double positives are highly selected for response. In fact, so much so that you actually got better response to the drug in triple-negative breast cancers in this biomarker positive population than what the drug graduated onto FDA with. So we had a response rate of 77% compared to the graduating arm of 55%, and about the same PCR rate in the control group. So Puma's very excited about this because we now have a completely new therapeutic pathway for a drug that was going to be going down HER2 positive. But we rescued it back to a different population of patients that actually need a new drug. And we have two specific biomarkers, phosphorylation biomarkers, that could call those women out. Nothing else but phosphorylation found these women. These are not HER2 mutated, these are not EGFR mutated, they're not HER2 positive. They're missed by all the current ways of measuring these women. And if we didn't have a protein modification-based assay, they would not have been found in the first place. So one of the things that we're trying to do is recategorize cancers across tumor types, but more at the pathway level. And we think that this will be a paradigm. We and others. This is not my idea. So a lot of people are thinking about this. We're just providing some ammunition in the gun to say, here's another example why this could work. So this idea of recategorization tumors by their activated signaling architecture could provide a very powerful, synergistic dimension beyond a genomics alone approach, and we're hopeful for this. And as I said, we have drugs that target specific proteins and their specific architecture, and we're now coming up with commercial assays that have those drug targets on there. So we even heard yesterday people talking about monitoring JAKs and stats. Well, we can monitor those even at the phosphorylation level on these assays as well, providing a way in the future, perhaps, actually a new way to integrate this data to the pathologists. It's part of a medical record, if you will, of that patient's tumor. In the future, maybe a pathway activation map will be generated as part of the molecular tumor profile of that patient, and the oncologist will use this for therapy. Now, I want to finish up by telling you a different vignette of how proteomics can make an impact at the clinic. And we're going to toggle now from the tumor to the blood. And this is something, obviously, that Larry's group and SomaLogic know a lot about, perhaps better than anybody, about what to look for in the blood and how complicated it is and how hard as shit it is. So one of the things that it's important to recognize is that, we have a long tail problem in proteomics, especially in blood proteomics. And that is that about 20 proteins or so constitute about 99% of all the protein mass in the blood. So this is actually taken from a paper from Lee Anderson. So this is all of the top proteins in the blood. And if you look at this green pie, this is like all the other thousands and thousands of different isoforms of proteins right here, which is a subset of this. And this is like, everything disease-related in that. So when you have SomaLogic's 5,000-plex assay, most of what they're measuring is in here. And so this is a problem because a lot of these things are obscured. A lot of these low-abundance proteins And I've seen some of Larry's slides, and it's like, you know, this long tail of abundance way out to the edge. You can barely see what the scale looks like of the amount of these proteins. Having assays that can measure these is very difficult because these high abundance resident proteins like albumin end up obscuring a lot of the things that you could see in the blood in these assays. Especially so in the field of proteomics, where a lot of discovery proteomics is mass spec driven and you're throwing blood into a mass spec and you're asking what's in it. You got to get rid of these proteins first. So what we wanted to do was find a way, a technology, that could help us drill into this low abundant proteome for our own discovery purposes. And we heard about Nanohydrogels yesterday. And I think in Steve, your talk, you kind of were talking about hydrogels. So we created these hydrogel nanoparticles because of their open meshwork structure and actually implanted in them bait molecules to help us suck up biomarkers, basically creating a little nano biomarker vacuum. And these nanoparticles do this by basically solution binding. And these nanoparticles are very interesting because of the surface areas of hydrogels. This is not surface binding. We can put in and impregnate these gels with all sorts of dyes that have very high affinities, and they're separated from the solution phase by a shell. And that shell can actually be tuned by size and by charge to allow things in or out. And what these nanoparticles allow us to do, depending on the bait, is to do something very helpful as an upfront sample preparation technique. So I can't draw the trillion of albumin molecules that be in a blood compared to, say, your biomarker of interest. I had an interesting, I asked one of my postdocs to try to come up with some visual that explained what a 10 to the 12th fold difference was, which is like the difference between the lowest abundance protein and the highest abundance protein. Because genomics is easy because the log fold difference is like 2-3-fold log order. Proteomics, it's like 10 to the 12th. Well, that's the same volumetric difference between the volume of Mount Everest and a virus particle, a single virus particle. That's 10 to the 12th. So he said to me, man, it's not a needle in a haystack. That would be easy. So, it's actually much more than that. And so what these particles allow us to do is to take, in this instance, the biomarkers of interest and suck them in and leave away these high abundance proteins, and we can actually volume exchange out. So imagine a blood tube of 5 mLs of blood and taking all the biomarker content of that and dropping it into a much smaller volume. Just by volume exchange, you get amplification. There's no PCR, but we can basically get that same end-up result by looking at the differences. And we can basically create a whole Baskin-Robbins flavors of dyes that all bind different types of proteins and biomarkers. And we can do this in all sorts of body fluids for all different types of applications. The whole point here is that by using these particles, we can basically increase the dynamic range because of the concentration effect before and after. And you can reach down farther into the level of the proteome. Now, I'm going to finish up by how we're using it quickly. One of the most growing infectious disease issues in the United States right now is the increase of tick-borne diseases. The CDC says it's the single most rapidly growing problem. Not Zika, it's tick-borne diseases. And the reason for this is that to this time, we do not have a test that's accurate enough to measure it. The advantages of a direct test are somewhat self-evident, but many people don't realize the current CDC test is an indirect assay, the test for the presence of an antibody against the tick protein. The test is so bad that the state of Virginia has a law they just passed about a year ago that says that a physician must tell the patient how crappy the test is that they're about to give. No lie, unbelievable as it is. The test has about a 50% sensitivity and specificity. I don't think I could get paper published where I said the end result of my work was a 50% sensitivity and specificity test. Yet that is the test today that you would get. So what we decided to do was to see if we could use these nanoparticles to form a direct test for Lyme, the direct test for the Borrelia spirochete protein. So a direct test has obvious advantages. You're looking directly for the presence of the bacteria protein as opposed to an indirect test where you're looking for blood antibodies that may or may not come up. So the failure of previous Lyme antigen tests have been it's too low in concentration to be detected readily. So we said this is perfect for our nanoparticles because we could concentrate this ostensibly and then see it with new tests. And that's what we did. So we actually found a nanoparticle dye that binds the Borrelia spirochete, the outer surface protein. And we actually tuned the dyes to bind to the peptide region that's shared across all the Borrelia spirochete species. The actual CDC indirect test actually only tests for antibodies against one of the Borrelia species. That's actually about 5% incidence. That's why it's really bad. So we just published the results of a clinical trial that's gotten a lot of publicity because we found extremely good sensitivity and specificity in our test compared to the matched CDC test. The most important thing out of this result, and the reason why we got a lot of publicity from this, was this right here. Persistent Lyme. These are patients that basically say they have Lyme disease, they think they have Lyme disease, they have all the symptoms of Lyme, but they go in and get the current test and it's negative. And the doctor says, you don't have Lyme, you have chronic fatigue, it's all in your head, you're anxious, you're depressed, you have ALS, you have all sorts of symptoms, neurological symptoms they get worked up for. In our trial, we found that 40% of those patients are positive for Lyme. 40% have the antigen in their urine. If you have Borrelia spirochete outer surface protein in your urine, you're infected. And this is the test that we actually have now on the market, and it's commercialized today. And the hope is, of course, that this type of approach can be used for many different indications. The Gates Foundation is funding us to develop a urine TB test, a malaria saliva test. And what we're hoping for is to try to change the dynamic where things that you could measure in the blood could actually be measured in urine or saliva if you could only measure down to a certain dynamic range. So we're working, DARPA has just funded us for a clinical trial. It's a little mini, it's not the trial that Larry described for the million people of Colorado. We're funded by DARPA to look at 10 people where we're getting their serum, saliva, urine, tears, capillary blood every week for a year. And we're going to use the nanoparticles to measure the immunome and to see if we can monitor immune function over time. And hope that we can measure that not by blood testing, but say in the saliva or urine. So we're hoping that this will be the result of a lot of work in the field of proteomics. Thank you very much. Appreciate your time.
Yeah, time for a couple of questions. What do we have?
Some over there, I think.
We have one there.
Presumably in the women with the triple negative breast cancer, the ones who respond to the drug, either the phosphatases are low or the ligands are increased. And I don't know whether you've done it or whether you would like to send those samples to SomaLogic to do it. But could you tell us whether you know anything about either of those parameters, either the phosphatases or the ligands?
So that's an excellent question, very prescient. We think that it's a ligand-driven event. We don't know which ligands. It's a hard question because there's so many ligands and there's so many isoforms of these ligands. We actually have a clue. We actually think it might be estrogen receptor. Those women that are high, they're triple negative, so they shouldn't have any estrogen receptor either. But this goes back to the current methods of measuring these proteins are inadequate now to explain some of the subtle differences. You may be negative or low, but there may be a high-low that is the real actor here. And in fact, this is what's occurring. All those patients in that upper quartile, they also have a statistically significantly higher level of ER. And ER is known in low amounts to act as a membrane-associated ligand, a non-genomic transcription factor. And so we think that this might be, but it's a postulate right now. And looking in the blood at these women, you know, through things like a SOMA array or some way to look at all these ligands, that's the way to go, I think, next.
Okay. Yeah, the lady there.
Are there HER2 positives that are phosphorylation negative, and are they not responsive, or is it just by having more HER2 that's sufficient?
That's a great question, and kind of from a time standpoint, I didn't get into it, but yes, what we're finding is that the women that are not responding, because it's imperfect, it's not like everyone with HER2 responds, of course, we're finding there's a negative predictor where there is no phosphorylation, and that's a whole separate talk. But yes, we think we can screen out those women that were HER2 positive non-responders by looking at phosphorylation.
Thank you. One, I think, lady at the back there.
Yeah.
No, we need a microphone,
Oh,
actually. It's coming. The guy's running up the stairs right now.
I'm fascinated by your Lyme direct antigen detection in the urine. Do you know for sure that you're not detecting spirochetes in the gingival crevice or other spirochetes in the urine? And is there a correlation with the level that you detect of that antigen in the urine with the severity of disease, disseminated Lyme, being higher than localized Lyme?
Yeah, no, it's an excellent question. So in the paper, which afterwards you get your card, I can send you the PDF, we actually mass spec sequenced the antigen that the nanoparticles are detecting, and it is the Borrelia spirochete common antigen that's in the urine. And most importantly, I didn't show it here, we have patients in the trial that were diagnosed by the test and got doxycycline, and then we were able to show with a direct antigen test that as their symptomatology resolved, their level in the urine went away, and it follows symptomatology.
Great. Thank you very much. We're going to move on now.